Hello, my name is Tina Hall. I'm a senior researcher at the National Food Institute of the Technical University of Denmark. This lecture will give you a short introduction to microbial risk assessment. But just to sketch the problem in short, there may be hazards in the food. And when we're talking about microbial risk assessment, those hazards are often pathogens, that is, microorganisms that can make people ill if they eat the food. And if these pathogens can survive, multiply, grow in the food or are transferred to other foods, then people get exposed to the pathogens and they ingest the pathogens through the food, and the amount ingested is called the dose. And then when you have an estimate for the dose, you can calculate or estimate the probability that people get ill by using a dose-response relationship, which is really a function of the dose and then the likelihood of becoming ill after ingesting the dose. So, but before we dig more into that, we will try to define some terms. We want to define hazard and risk. Often hazard and risk in the general population is used interchangeable. But in the context of risk assessment, they are in fact quite different. So a hazard is like a physical thing. It's a biological or chemical, could also be a physical object, that are capable of causing adverse health effects, illness or damage or harm on the human body. Examples of hazards can, for instance, be bacteria, viruses, parasites. So the biological hazards. Within the chemical hazards, we can have environmental pollution, such as heavy metals. We can have allergens. We can have different toxic compounds that are formed under or during cooking. And then finally, we'll have the physical hazards, which could be broken glass in the food. It could be pieces from utensils in the food as well. So this is the hazards. Then we have the risk. The risk consists actually of two things. It consists of the likelihood or the probability of a hazard causing adverse effect on the consumer, but not only the probability of illness or adverse effect, but also the severity or the magnitude of the severe effect or the adverse effect. And if you look at that in a more, you could say, equational way, you can say that risk is a product of the probability of occurrence and then the severity of the adverse effect. So you have here the risk is a function of this probability of occurrence and the adverse effect. And if either the probability or the adverse effect that it causes is 0, then the risk is 0. But in the general rule, there is no such thing as a zero risk. There will always be a risk. It might be extremely small, but it will usually always be there. So having defined these two terms, hazard and risk, please pause the video and think about what is the hazard and what is the risk in this cartoon. So let's see if you got it right. The hazard in this is the snake. Or it can be more detailed, it can be the poison if it's the poisonous snake, if it is a poisonous snake. But the fact that it is something physical that can do you harm. The risk, on the other hand, is like a chain of conditional probabilities. It is a probability that the snake escapes from the terrarium, that it actually locates you and it bites you. And it is also the probability that you will then, after being bitten, become ill, and how severe your illness will be. So, taking you through the steps of risk assessment, there are 4 main steps: hazard identification, hazard characterization, exposure assessment, and risk characterization. As you can see, we also put the first bullet point here to the left in, which is called statement of purpose. It's not really an official part of risk assessment if you look in kind of documents from for instance, the World Health Organization or from Codex. But based on experience, we like to put it in because it is very important that the purpose of a risk assessment is stated very clearly. And it is actually the risk managers, those that are going to manage the risk, that should ask the very clear question, what is it for a risk that you want to assess? What is the questions you want answered? Otherwise, you could end up making a lot of work on providing answers for something the risk management didn't really want it answered. And therefore, the statement of the purpose of a risk assessment is very crucial. The hazard identification is kind of very obvious. It can be a microorganism, it can be a toxin, but it's this physical thing that are able to cause adverse effects, illness in humans. Very often it's given beforehand. The risk managers will know exactly what the hazard is and what they want a risk assessment to involve. But it can be that risk managers would really like to have an assessment of all risk occurring in a particular food. For instance, what is the risk? What are the hazards occurring in green salad, and what is the risk then of consuming green salad? Then you will first need to identify all the hazards that can occur in green salad, and then they make a risk assessment based on these findings. The hazard characterization is then either a qualitative or quantitative evaluation of the severity and duration of the adverse health effect, illnesses, that are associated with ingesting the microorganism. This can be a quite complicated step because it needs to consider both the microorganism, the host, and the food and the factors related to that. For the microorganism, it will often be the ability of the microorganisms to replicate or growth in the particular food. It can be whether the microorganism has special violence or infectivity traits that you need to consider. Whether they can transfer genes. It could, for instance, be antimicrobial genes, if it can transfer genes from one bacteria to another. Also, whether there is a great chance of secondary transmission. With secondary transmission, we mean spread to other humans. So whether how contagious it is for the transmission between humans. And then also something about persistence. How persistent is it in the environment? It could be in a production environment on a slaughterhouse. How likely is it to survive outside a host for a long time? The host, which when we talk about food safety risk assessment, is the human body. Again, genetic factors can play a role. Genetic factors can influence the susceptibility of people to become ill. Other underlying diseases can also affect susceptibility if it reduces your immune response. A previous history of exposure to the same pathogen may also alter the susceptibility, but in the opposite direction, because you may then have some kind of immunity to become infected with that particular microorganism. And then finally, there's the food. The attributes of the food can also affect how infectious the microorganism is. There are studies that show that if the food has a very high fat content, then the microorganism has a tendency to hide, you could say, in the fat, and then pass through the acidic environment in the stomach and reach the intestines quite unharmed. And that will then give rise to more infections or more severe infections. It can, of course, also be the other way. So if you somehow have treated the food with a high salt content, for instance, then you reduce the probability of growth and survival of the pathogens in the food. Based on this, you may be able to determine a dose-response relationship. Often this is something you find in the literature, in the scientific literature, when you're doing a risk assessment, because it's studies that has been done. It can be actually human feeding trials that are estimating for each dose level how many people get symptoms, the proportion of people getting symptoms, and how severe the symptoms are. Of course, there are not many human feeding trials available because there's some ethical things here that needs to be considered. So dose-response relationship can also be formulated based on outbreak investigation. So if you have an outbreak with a foodborne outbreak with a certain pathogen in a certain food, and you know how many people that's been exposed, and you then know how many of these that have become ill, you can then also estimate a dose-response relationship. Then you have steps, then you have the exposure assessment. The exposure assessment is really a description of the pathway. So it's a description of the particular food that you want to investigate and what happens to the food through the production process all the way to consumption. The purpose of the exposure assessment is to estimate the dose, to estimate how many bacteria or microorganisms are the human consuming at the point of consumption. It can also be exposure from other sources. For instance, it could be direct contact with animals or direct contact with other humans. But when talking food safety, it's usually through the food that people are exposed. This needs to obviously focus on estimating pathogen prevalences. So if you have a food, how many percentages of this particular food is contaminated, packages of food, for instance. But also what is the concentration of the pathogens in the food, how many microorganisms per gram of food, for instance, is present. Then we need to, because this measure will often take place at some particular point in the production. It can be the end product, but it can also be before the end product. And then you cannot be sure that this will actually also be what is present at the time of consumption. Also, often people cook it or prepare it and something will happen to the food. So we need to estimate the change over time through the different processes from the measurement we have and on to consumption. And for this we use predictive model. Predictive models are those that are estimating growth or inactivation of bacteria through the process, based, for instance, temperature, storage time, different physical or chemical factors that are present in and around the food. And then finally also consumption patterns needs to be considered. We need to know how much people are consuming, and sometimes it will be also useful to divide that up on age group or gender groups. Finally, we have the risk characterization. The risk characterization is really integrating the information from the 3 previous steps, particularly hazard characterization and the exposure assessment. So if you remember, the hazard characterization was something about formulating the dose-response relationship, and the exposure assessment gives you the dose. And if you combine these two, you will get a risk estimate, which is the probability of human getting ill from eating this particular food. The expression of risk can be done both qualitatively and quantitatively. Qualitatively is very hazard focused and it is descriptive, and it's typically used if you are short of time, you need to provide a risk assessment very quickly, or if you are really lacking data. It is not as objective as a quantitative assessment. You describe it in terms such as low risk, medium risk, or high risk, but then again you all can ask yourself what is a low risk, what is a medium risk, and so on. So if it is at all possible, then it is better to try to describe the risk in a quantitative way. Because this is risk focused and it is a numerical and more objective terms. And often we will describe it as a probability distribution. In this example, it could be the probability distribution of the amount of food that are contaminated at the point of consumption. You might have data on this. You might have 10 samples, as in this example, where 2 of them were positive. The most obvious answer to this would be yes, then we have a 20% prevalence rate. But the thing is, with only 10 samples, we have a very high uncertainty on this estimate. and so really the true prevalence could lie anywhere between almost 0 and up to 60%. So therefore, including uncertainty and also variability in your risk estimates are important for the risk managers to actually make good decisions. So finally, to make good decisions, you need to consider as many risks and as many factors as possible. But also remember that this risk assessment model is only a model, and it will never be able to describe the reality in full. So it's the best guess on what the risks are. Thank you.